Hello, everyone, from the COVID-19 Clinical Research Coalition. Good morning, good afternoon, good evening, wherever you are, and a very warm welcome to you. That is funders, researchers, and our allies. We are attempting this evening to address neglected COVID-19 research priorities in low-resource settings. I'd like to just start with a couple of housekeeping slides, if I may. Next slide, please. So this will be recorded and the link will be shared on the website. If you've got any technical difficulties, there's the email. After the meeting, you will be asked to fill out a short survey. Many thanks in advance for giving us feedback on this event. And if you're a tweeter, please use the COVID-19 CRC at COVID. You can see I'm not a tweeter, to tweet about the coalition and this meeting. Next slide, please. So during the Q&A, please raise your hand in the normal Zoom way, and then the moderator will call you. But you can also write questions using the Q&A tool. That's at the bottom of your screen, and you can comment and upvote people's questions by clicking on the thumbs up icon. We're very interested, of course, that's why we're having this meeting, to hear the funders and your views, and we very much value that. So we look forward to a very good discussion. If we do run out of time in the Q&A sessions, please provide your comments or your questions on our comments, questions, and your interest in project participation form, which will be posted in the link in the chat after each session. And we'll try and answer all these questions, even if we don't get time at the online, as it were. Next. So quickly, this is the agenda. This meeting is really to discuss four priorities, or what our coalition have considered after a long process, which we'll explain to you, to be four priorities for support, and to discuss why these should be supported and how that could be done. And we're very grateful also to have some distinguished contributors. Professor Dr. Veronika von Messling has very kindly agreed to open the meeting, and then at the end we will have a talk from Dr. Soumya Swaminathan, who is, as you know, the Chief Scientist at WHO, and then closing remarks from Charles Wisonge. So next slide, please. Very briefly, this is us. We began about 15 months ago, and we came out of really a broad concern that the research needs of peoples and health systems in low resource settings were not being addressed adequately or prioritized in the global response to the pandemic. And we've seen the consequences of this over the past year. And also worry that the impact of COVID on already fragile healthcare systems could reverse some of or many of the gains that have occurred in the past decade or so. Since we started, the coalition has grown quite substantially from the original 77 signatories in the Lancet comment that launched us in April 2020 to more than 200 institutional members and nearly 300 individuals, representing overall 90 countries. Next, please. So this is just a very brief snapshot of a lot of work that goes on in different areas. I'm not going to go into it in any detail, but just to say that the engine of our activities are the working groups, and it's the products of these working groups that, or four of their products, that we'll be discussing tonight. Next, please. This is a map kindly produced by our colleagues in UK CDR showing who's doing what worldwide. And you can see that not a lot has changed, and most of the research is going on, unsurprisingly, in resource-rich areas. So, as I mentioned previously, several of our working groups, or all our working groups, have taken a bottom-up approach to trying to identify what they consider to be the very top research priorities in low-resource settings. Thank you, Leila. And today you're going to hear the four, what we consider to be the four leading candidates, but that's not all of them. And we're fully aware that that we're not the only people to have set priorities, and we know that these priorities have been identified by others, and they're consistent with the WHO's R&D blueprint, for example. But we feel that the level of commitment to support these areas is not really commensurate with their importance and their impact in low resource settings. It's not a naked call for funding. Obviously, it's a discussion with you. So ultimately, we hope these things will be supported. But it's an opportunity to discuss them and hopefully promote them, and also to learn from you, the funders, and others on the call to advise us so that we can be constructive in moving forward. So the best outcome really from today would be that the information that you hear from us helps you shape your funding calls in the weeks and months to come, and that the information that you hear or that the investigators hear will help them strengthen their proposals. So that's enough from me, and I'm going to now try and turn myself off. And it gives me very great pleasure to introduce Professor Dr. Veronika von Messling, the Director General for Life Sciences at the German Federal Ministry of Education and Research, known as BMBF. This has been a very strong supporter of our coalition since its launch, and it's funded our secretariat, for which we are enormously grateful. BMBF is supporting many initiatives related to the pandemic, among them the anti-CoV trial, the largest global trial in early treatment of COVID, COVAX, the ACT accelerator. So these are the main pillars in our research fight against COVID, and various national projects too. So I'm very, very delighted, and I'm sure that Professor Dr. von Messling will tell us more about their priorities in this area, and we're very delighted to hear from her. So over to you, Veronika.
Thank you so much, Nick. Dear colleagues, and thank you for inviting me to give an opening address at this meeting. I'm honored and happy to speak about the particular importance of supporting research in all countries affected by the COVID-19 pandemic. Research and development, innovation, and access are essential drivers for health and well-being worldwide. The German government, and in particular our ministry, have pursued the goal of building research capacities and ensuring equitable global access to much-needed therapeutics, vaccines, and diagnostics already for a long time. We are committed to fund global health research and development because we firmly believe that such funding will significantly advance global health as a whole. We delivered on this promise for the COVID-19 pandemic, as this crisis affects all, including the world's most vulnerable populations. This is also why the German Research Ministry immediately supported the launch of the COVID-19 Clinical Research Coalition last year. I'm absolutely convinced that only such a coalition will bring collaborative solutions to the COVID-19 crisis, especially in low-resource settings. It is essential that this forum provides the opportunity for open discussions between committed researchers from low and middle-income countries and their allies from the North, as well as funders and donors. This open exchange is essential to deliver on the vision of the coalition to have a global research response to COVID-19 driven by the needs of low-resource settings. So it is clear that this coalition will not work without you and the efforts of all your devoted researchers and clinician colleagues out there, especially in hospitals in low-income countries. I highly appreciate your engagement in the fight against COVID-19 and also the input that you bring to the working groups of this coalition. As you may know, our ministry funds product development partnerships, PDPs, for about 10 years now. PDPs, like DNDi, jumped into the gaps either left or never really addressed by private industry. Since these early days, PDPs have developed urgently needed treatments rapidly and successfully, but there is still need of urgent action. There's another reason why we decided to support PDPs. PDPs are also strong advocates for equitable access worldwide and work for the goal to leave no one behind. I take pride in saying that improving access for everyone is one of the guiding principles in the German Research Ministry for supporting funding initiatives in global health research. An example is our funding initiative, Research Networks for Health Innovations in Sub-Saharan Africa, which clearly addresses local health needs. This initiative not only helps to improve public health in Sub-Saharan Africa, but tackles one of the major problems in global health research, the fragile and often even lacking research capacities in low-resource settings. The funding initiative has three main objectives: supporting African academics in leading international research consortia, providing junior researchers with opportunities to develop research skills, and the transfer of research findings into policy and practice. Coming back to the COVID-19 pandemic, I think we can now see a light at the end of the tunnel, or at least we hope so. Following a rapid reaction to the spread of the novel coronavirus, vaccines against COVID-19 were developed with unprecedented speed. Here, the immediate response of the Coalition for Epidemic Preparedness Innovations, CEPI, played a crucial role. CEPI was founded in 2017 in the aftermath of the Ebola outbreak in West Africa, with the mandate to develop vaccines against emerging infectious diseases, which mainly occur in tropical regions. Thankfully, this included platform technologies for an unknown disease X, which hit us over a year ago. The pandemic has shown us that with this initiative, Germany and other funders have paved the way for better pandemic preparedness. In the COVID-19 crisis, we also support the Access to COVID-19 Tools Accelerator to secure the development of and the equitable access to COVID-19 tests, therapies, and vaccines for people across the world. Global joint action is mandatory to overcome the pandemic. And you heard so many times already, but it is still true, this crisis will only end if it ends for everyone. We already made important achievements in overcoming this pandemic, but still many knowledge gaps remain. I look forward to open discussions today and hearing about your experience with COVID-19 research in your countries, your ideas for cooperation, and your strategies for the future. Thank you very much.
Thank you so much. That was a perfect introduction to this meeting. We're very much appreciative of, and you set the tone perfectly. So now I'd like to get to start with the first of our sessions, which is the characterization of long COVID or the post-COVID condition in low and middle income countries. It's my pleasure to introduce Professor Juan Carlos Villar. He's Clinical Epidemiology Working Group co-chair in our coalition and Professor of Medicine at the Universidad Autónoma de Bucaramanga. He's also Head of Research Center at the Fundación Cardioinfantil Instituto de Cardiología, Bogotá. Apologies for my Spanish. He will speak to us about the research project that they've been developing, focusing on post-COVID in low-middle income countries. And to moderate this session, I warmly welcome Dr. Gail Carson, my colleague on the steering committee of the coalition. She's the Director of Network Development at the International Severe Acute Respiratory and Emerging Infection Consortium, known as ISARIC. She also leads GLOPIDA's Scientific Secretariat and is the Vice Chair of the Global Outbreak Alert and Response Network, which is GOAN. The floor, Juan Carlos, is yours.
Thank you very much, Professor Wright. And it's a pleasure to be here and share with you, colleagues, these thoughts on behalf of the Clinical Epidemiology Working Group of the Coalition. We are all concerned on long COVID as a concept, but as a scientist, we are also concerned on the dimension and characterization on what is long COVID, what it really means, and how much we can see of it and how much is attributable to it. Okay. Long COVID is a wide spectrum of persistent and emergent symptoms following COVID-19, but the construct is still ongoing, and there is varying definitions and a process of ongoing characterization by the WHO and others. And most importantly, studies are and control case series, so they are actually ranked under low certainty of evidence, and they have mainly hospitalized patients. So we don't have precise, reliable estimates, and the information is still uh, unreliable that way and inconsistent in terms of the numbers. The map of the COVID-19 research tracker show us that although we have around 70 long COVID projects by April 2021. Uh, the subpopulations that those projects include are not specified. There are varying populations, as I said, they're mainly hospitalized patients, and the trials in vulnerable populations are very, very few. And the world map, as Professor Nick just show us many projects are on the spectrum of long-term symptoms, still ill-defined, but for the most part, the research is conducted in upper high-income countries. So there is a need for embarking on an ongoing validation on construction of the definition on long COVID features. And we definitely need a more reliable and precise definition estimates of incidence, especially derived from comparisons from non-infected controls and also outpatients. We need inclusion of broader populations and especially underrepresented populations in what I'm calling here special cases, special clinical cases, or special populations with particular social needs, and particularly including populations from Latin America, Africa, and Southeast Asia. And also, we need to complement the perspective on a wider way, including healthcare services research, social research, qualitative needs, and aspects on these populations. So in our group, in the committee of the coalition, we made an exercise of prioritizing questions, and we end up having focusing on a question on that became our goal to define and assess the magnitude and diversity and also social and economic impact of long COVID outcomes, and under the umbrella of the coalition, to devise a strategy to, on one hand, to partner with the ongoing international registries such as ICERIC or the WHO Long COVID Global Initiative, or the clinical trials such as anti-COV that are ongoing in Africa, as in terms of logistics, data sets, collaboration, networking, etc. And on the other hand, to add value to our clinical research by including other fields, and going beyond that and studying special populations from lower and middle income countries. Everything to um, end up in conducting a unique international prospective clinical study that has at least 12,000 participants from the continents that I just mentioned, followed for a couple of years, and including coordination sites in each continent, and also, why not, to set up that structure to do potential work beyond the pandemic. So the study design that we are thinking of, it has an inception and with retrospective data collection to reach what happened in the infection times, and a data perspective follow-up to gather data on 1 year incidence on outcomes, and working in the along the way, in an ongoing and improved and validated definition of long COVID outcomes, and also, why not, do extended follow-up, enrolling On one hand, what I'm calling here as selected populations with previous SARS-CoV-2 testing, gathering seropositive and seronegative individuals, including seronegative individuals that are discharged from hospitals, and also to enroll a variety of special populations. This is just examples of important populations that are underrepresented and need to be studied with to make valuable comparisons between seropositive and seronegatives, and also among the special groups of seropositives with the general population of seropositives. The study's main goals would be to generate data on 1 year incidence of a variety of outcomes, to study the variations on healthcare access and delivery, to provide information of prognosis factors by comparing infected and non-infected populations, and also by comparing, as I said, among the special and unselected populations that are infected, and also to enhance the value of the study by doing context-specific social and economic studies. The way we see it, this project will have 3 coordination sites, 1 for each continent, and hopefully there will be at least 3 countries by region. We think of enrolling at least 6,000 people of the so-called unselected population to study outcomes that are as frequent as at least 3% in the general population, or 10% in the special populations. We are aiming to provide information on clinical outcomes divided into categories like this: cardiorespiratory, hematological, neurological, mental, etc., and also to provide information on well-being, quality of life, and also the delivery of healthcare resources and health economics, etc. We think of a timeline for 2 years, where conducting a pilot study in the first 6 months, setting up the steering committee, subcommittees with the coalition, and having our first patient in the last center at year 1, and having at least 24 months to have our last 1-year visit. And we will take the lead as a committee of this study, but also gather the work and the collaboration with many other colleagues from the other committees of the coalition, especially with the data sharing to do data administration, a lot of data transfer, etc., but also the other committees that you are seeing in this screen. And just to wrap up, what is so special about this project, in our view, is that it will represent the underrepresented in terms of both countries and populations. It will generate valid definitions and incidence estimates especially by the inclusion of outpatients. So we know really what is the incidence. We don't over or underestimate what is long COVID, and also by including non-infected inpatient control, so we know this is a condition that is really attributable to COVID and not to being in hospital, etc. What is also special of this project is the possibility to go beyond clinical information to integrate biological, clinical, and social economic aspects, and most of all, to do the much needed South and South collaboration, both within this pandemic and beyond that. So thank you very much, and sorry for that interruption along the way. Thank you.
Thank you very much, Juan Carlos. That was absolutely fantastic and very clear, and super to hear about some really thoughtful consideration given to controls, something that I think for many of the studies trying to characterize long COVID have been missing. So we do have one question in the chat so far, but I'd like to take the opportunity as moderator to ask you one, and to give our participants an opportunity to add more questions to the Q&A. Please can I ask if someone's not presenting, please mute yourself. Thank you. So Juan Carlos, I think one of the reasons, and you cited it yourself, why you feel and your group feel this work is so important is around helping it give and provide data and information to policymakers and those that strengthen healthcare systems. And we know that the pandemic has indeed toppled these, unfortunately, in some of the low and middle income countries, and certainly severely dented and threatened other systems around the world. So how do you actually see the feeding in of the outputs from this study to actually help strengthen healthcare systems in the low and middle income countries?
That's a very, very important question, Gail. Conducting a clinical study that has a sort of a standardized way to both gather the information and following patients will work as a railroad to give some structure to handle these the populations that are included, and hopefully to expand that structure to healthcare services as a reference. It is well known that clinical trials and cohort studies work by within the study population, but also by having a sort of a halo to be a reference for standard care in patients. And obviously, we expect that patients, while followed, are taking care in the best possible way with the standard of care. And the project by itself is a way to gather information from outside the study to make the most up-to-date type of clinical care, and hopefully to communicate it along the way to healthcare authorities and regulators to make the best possible way to manage and to give or provide care to these patients.
Yeah, no, I think that's a super point, and something that's so important when we have what seems to be a multi-organ disease that may actually cause symptoms for these people for months, if not years, and probably will continue to stretch our healthcare systems because of that multi-organ insult. We've got a question in the Q&A from Soraya Gazi, forgive my pronunciation, Dr. Gazi, from Fiocruz in Brazil. And Dr. Gazi is asking, I would like to know how you will address the different SARS-CoV-2 strains, considering they have different infection rates and probably different symptom intensity. Carlos?
We are first of all, we are just thinking still in broad terms for this study design, but for sure this is going to be a very important point. And the beauty of this idea is to have very different populations and to take advantage of the opportunity of having many different strains of the virus infecting population, and definitely relating the strains with the clinical outcomes, both in the acute phase and the long-term phase, for sure.
Yeah, so it's going to involve a lot of close working with our laboratory colleagues.
Yeah.
I'd like to give you another question from the Q&A from Naomi, who's asking which countries are part of the study so far. Have you got that secured yet?
You have as starting points. We have the ongoing registries, international registries, where there are countries participating, but we have to first decide on the continental coordination sites and then by working within the coalition to end up with a list of countries and especially a list of sites that are included in the study. The goal would be to have roughly 30 sites, 10 for each continent, and not to burden very much each site, having a commitment of including roughly 400 to 500 patients, half and half of the so-called general population, and selected populations that are easy in terms of logistics to gather for that particular site.
Great, thank you. And we have a question from Robert Colabunders, who's asking, do you already have a data collection tool that could be used internationally for this?
No, not us particularly. We are just in a starting point, and we will have to along the way to agree in a particular data collection points, but we don't have that yet.
And there may well be opportunities there to work with us at ISARIC, and we're going to be exploring
Absolutely.
that together.
Absolutely.
So we're looking forward to that discussion. Got another question here, if I may. I think we've got about 3 minutes or so left. How can we contribute to the part of the active research to include a country like Nepal into the study? And that's from Gaurav.
Sorry, I didn't get it exactly.
I think Gaurav's asking how could Nepal, for example, contribute some sites if they are not already on your radar?
Oh yes, I'm thinking exactly on countries such as Nepal, in Southeast Asia. It definitely would be a challenge to decide on India as a big country that is already in some of the international studies, or other countries, neighbor countries that are underrepresented, and Nepal is one of them for sure.
Great.
Yep.
And we've got a question from Christian, who's asking, Juan Carlos, please can you describe how people could actually join the study? How do they find out about this when it's launched? How do they get involved?
Okay, thank you. We have the website of the coalition, and within the coalition we have the clinical epidemiology working group, and there is a contact link for that. And this would be the primary way to contact, and we'll definitely get back to you. And we are very interested in having new members in the committee on one hand, and also very keen on providing opportunity to colleagues from different locations to join.
Super. And Natalie would like to come in here.
Yeah, thank you very much, Gail and Juan Carlos, of course. So, no, I just wanted to make the point and re-insist maybe on the, the unicity of this project, which will allow a lot of information. The idea is to get, I think, about 12,000 patients in that specific project, 9,000 of which would be infected. It will be both retrospective and prospective. If the data from this study are shared, and I think we need to insist on this, with the other projects, such as the big cohorts that ISARIC is conducting and others, then yes, we will be able to respond to all the most, we hope, of the questions related to variants, to subpopulations, etc. Because the event rate is not so high, luckily. I mean, that's a good news. But if you have 10% and the epidemiology working group has been looking at this calculation. But if we want to go for more subtle information and maybe look also at the impact of treatment on preventing long COVID and many other things, I mean, this is a project that is key, especially also because it's the only one that's looking at this for LMICs. So we really need to, I think we need to help and contribute. And I'm taking another hat, which is a hat of a clinical trial that's running in Africa for now, and maybe also in India, anti-CoV. And the epidemiology group has asked if we would be willing to contribute with a follow-up of the patients enrolled in the study. And of course we would, and we are very much in favor of those a collaborative project. Over.
Thank you very much. I look forward to discussing it with both of you. That's our session over. Thank you very much, Juan Carlos.
Thank you.
Yes, indeed. Thanks, Juan Carlos, and thank you very much, Gail. On the question of if you'd like to participate or get more information or partner with this group, there's a form that the Secretariat will share on the chat, as well as the other the more conventional approaches to the coalition. So up next, it's Dr. Wilber Sabiiti. He's a member of the coalition's Virology, Immunology and Diagnostics working group. And Dr. Sabiiti is a biomedical research scientist and the senior research fellow in medicine at the University of St. Andrews. Wilber will present a research project developed by his working group on biorepositories and sequencing. And to moderate this session, I'm very glad to welcome Dr. Divya Shah. Divya is an immunologist, but she's also the epidemics research lead at the Wellcome Trust. Over to you, Wilber.
Thank you, Nick. Next, Damian. So I'm representing the Virology, Immunology and Diagnostics Working Group. We are 16 members. We cut across continents, and over 90% of the members are from the Global South. And we are still recruiting more members to join the group because we believe they will be crucial in undertaking this project that we are proposing. Next. So biobanking is a systematic preservation of biological samples for future use. The importance cannot be overstated. Immediate COVID-19 clinical and public health response, load of work is immense, which means that the people responding cannot be able to answer all questions at the same time as responding to patients and managing other public health issues. So biobanking is important. And when you have biobanked samples, then this is important for development and optimization of novel diagnostics, vaccines, and therapeutics. Whole genome sequencing is a comprehensive mapping of the organism's genetic information. As you may all know, the early publication of SARS-CoV-2 genome was cornerstone to development of diagnostics and vaccines that we have today. Yet this kind of capacity is still missing in many low and middle income countries. So having functional whole genome sequencing capacity is critical for early detection of COVID-19 variants. As you may have seen, many variants have emerged, and sequencing has been crucial in doing this. But we still have a shortage in the LMICs in terms of detecting these variants. The earlier the detection of variants, the more accurate will be the tools to respond to the pandemic. Next. So what are the challenges based on the existing capacity? So in the biobanking space, there are difficulties to access these samples. So requests often for specimens go unanswered, meaning that even though there may be some samples somewhere, they may not be easily accessed. Often specimens are not fit for purpose, means they have been preserved, but they are not suitable for the questions being asked. The governance and ownership is a challenge because often these samples are owned by individual academics who are less, or they are not of a public resource. So they become quite hard to access. quality is not guaranteed, and because it's not clear how they were preserved and whether they've gone through cycles of defrosting. There is paucity of inter-biobank standardized biobanking protocols, and this needs to be addressed. And varying regulatory and ethical framework for what should be considered for biobanking specimens. In the space of whole genome sequencing, there are infrastructural challenges, especially equipment, computing power, and unstable utilities such as electricity. It's inadequate bioinformatics human resource capacity to interpret genome sequencing outputs. And then procurement challenges, which covers affordability and timely acquisition of sequencing materials. These are the challenges, but if we come together, we can overcome these challenges and ensure that this capacity is available in LMICs. Next. So the aim is boosting scientific response to COVID-19 in LMICs by increasing biobanking and whole genome sequencing capacity in LMICs. Our primary objectives is establishing and or enhancing biobanking capacity in LMICs, enhancing genome sequencing capacity for LMIC biobanking centers, and streamlining biobanking and sequencing methodologies in LMIC. The second objectives are building partnerships with key institutions, stroke agencies in the biobanking and genome sequencing space, conducting responsive research on evolution of SARS-CoV-2 and its effect on COVID-19 diagnostics, the vaccines and therapeutics, and then advocating for biobanking and data sharing regulatory framework that is fit for purpose. Next. So this is how we want to approach it. We have reviewed several ways of establishing this capacity. There's a centralized model, which basically has one center and then different sites worldwide sending samples to this center. And then there's a regional model where you have maybe a number of countries that are sending samples to one regional hub. Then number three, the decentralized network model, where all samples are collected, characterized, and stored at the site of collection. We believe this is the best model, whereby it could be for smaller countries, maybe one biobanking and whole genome sequencing center, or for larger countries, 2 or 3 centers in such a country. The advantages are it obviates the need for specimen transport across national borders, which is quite complicated because if you do this if you want to do this quite quickly, it's not easy because you have to go through many negotiations. It's less costly, it builds local capacity, and is more sustainable. It allows ample time to negotiate material transfer agreements if necessary. And there is a Zika plan virtual biobank network, which we can draw on in terms of networking these biobanks virtually. Next. So we cannot biobank or preserve all samples. And if you just save samples without having a purpose while you're saving them, you're likely to save samples which are not fit for purpose. So for that, we want to make sure that the biobanking and WGS are kind of research question guided, which means this will answer the fit for purpose question for the preserved specimens. It will streamline the purpose, the number and type of specimens to be preserved, and it will guide minimum pre-biobanking information to be collected about the specimen. Some of this information is, for example, time of collection after diagnosis, vaccination status, gender, comorbidity status, disease severity, etc. So you can only collect such information when you have a purpose. So the sample you preserve, it has value. Some of the questions that we think we can ask in terms of research, what are the emerging COVID-19 variants? And these can be detected either as surveillance or as part of research. which form of the SARS-CoV-2, either wild type or variant, underlies breakthrough infections of vaccinated individuals? What impact do the variants have on the vaccine efficacy and diagnostic accuracy? What are the genomic and immunological characteristics of long COVID? You've already had the presentation about long COVID, and we will be supporting that working group in terms of answering this question. And what is the effect of underlying diseases like HIV, malaria on vaccine-induced immune response? Next. So we believe all LMICs need to access this kind of capacity, to build this capacity. So this kind of network, you see, is just representative of the aspiration to cover as many LMICs as possible. And we hope we can establish one biobank and sequencing facility per LMIC. And for large countries, maybe considering some subnational level facilities. Countries must express interest to co-partner, because this is very important, because if a country is not interested, it becomes like an imposition if you just bring the services to them. We want to leverage on the existing biobanking and sequencing facilities in the countries. Regional coordination centers in collaboration with the coalition secretaries will help to work with and leverage on finds biobanking expertise and WHO Africa sequencing network. Create a platform for North-South and South-South knowledge exchange, and work with the coalition's working groups on regulatory ethics and data sharing work packages in order to enrich the kind of work that we will be doing.
1 minute.
Note, as per find experience, setting up fully functional biobank in LMIC may cost around US dollars 150 to 500K. Next. So these are the roles of the member sites, so the country biobanks and sequencing facilities. We want them to take the lead in giving the services. And then the regional coordinating centers, they will be coordinating the member sites in terms of meetings or lobbying, or making sure that protocols are standardized. Next. This is how we want to address it in terms of operational or implementation arrangements. We will have a scientific advisory board, a steering committee made of representatives from sites and also from regional coordination centers. Next. The timeline, we think this would be like a 5.3-year effort, and the 3 months will be for proposal stage, whereby we want to make sure that we save time, because COVID evolves so fast. So you want to make sure that some of the things are done before we want to start as quick as possible. So 3 months at proposal stage, and we agree on the protocols which can be implemented as soon as possible. So, and research will go on from month 7 until the end of 5 years and answering different questions. We believe we shall deliver established biobanks and sequencing facilities to respond to COVID-19 and other disease outbreaks. We will offer comprehensive research that fills the national and regional data gaps on COVID-19 evolution and its implications. And then we will have national and international regulatory guidelines on biobanking and genomic data sharing. Next. Thank you very much, and I welcome your questions and comments.
Thank you, Wilber. So if I open it out to questions from the floor, if you'd like to put questions in the chat or raise your hand. We have actually got a couple of questions coming in the chat. So if I put those to you first, Wilber. So actually, this is one of my questions from Juan Lee. Who is the head porter to care for this biobank, specifically data?
So we believe, so members of the working group who come from these different LMICs, and most of them are academics, so others are working with public research institutions. They will be responsible for setting up or supervising these biobanks and sequencing facilities. And that's why we are trying to recruit as many more people into our working group so that we have representation on almost every LMIC.
Great, thank you. I guess you've mentioned the methodologies you discussed, so the decentralized models and the other models. So what's the kind of, I guess you mentioned some benefits of the decentralized model, but have you given consideration to perhaps a hybrid model, for example, decentralized and the regional model?
Yeah, we've given consideration that we had a good conversation with FIND, who FIND has been able to establish, I think, 4 biobanking centers across the world. And I think in Africa, there's one which only exists in South Africa. And as you may see, that is a really big regional kind of center. And I don't think it is even collecting samples from outside South Africa. So the regional centers will be considered based on the level of collaboration that exists between the countries. For example, if you talk of the East African community, if there's an agreement in those countries that samples can move easily in such a regional block, then a regional hub can be effective. But where there is no such regional close collaboration, then maybe a country level kind of coordination is more effective.
Thank you. There's a couple more questions, actually got quite a few. So from Irene Joao, how do you plan to deal with ethical issues that arise from this kind of research? Great question.
That's a great question indeed. And as part of the working packages, the work packages for the project, there is the ethics and regulatory framework for biobanking and sequencing. So data sharing and what are the issues. There are still many concerns, especially LMICs, why samples should be stored for a long time. And we believe that national level teams will work with their national level ethics committees to put in place the guidelines of what should be considered for samples or to be biobanked or to be sequenced, what are the matters of consent and how they should be dealt with. So national level, we will be really banking on our national level teams. I believe dealing with it at the national level is quite crucial because it all starts at the national level before it goes to the regional level.
Great, thanks.
But we will work with the ethics committee, the ethics working group in resolving this matter that will be guiding us.
And I guess importantly also to have representation on your scientific advisory committee as well.
Yes.
Okay, so another question. So a question from Professor Nick White. Should provision for biobanking virus samples, if feasible, be included in all epidemiological and clinical studies?
Yes, I think that actually makes it more purpose-led, because if a question is asked in the clinical and epidemiological aspects, then this can be integrated so I believe our research in a way from the different working groups can be integrated so that we don't have to duplicate effort.
Yes, and I guess sort of also related to that it's important, sorry, actually I'll come on to another one. Irene Joao, so she's got another question on the need for this research. It needs maybe some quite intensive community and public engagement. So how, you haven't addressed that, I think, in your presentation. Would you like to address that now?
Thank you. I think that's a good point. And I was trying to fit everything in 10 minutes, but community and public engagement is very important because it again speaks the ethics, the ethical concerns that the public has about preserving their specimens, any issues they have. So I think, yeah, and also buy-in because they need to know what is going on with their samples and get feedback from the research. So yes, community and public engagement is a work package that will be integrated in this.
And again, similarly, including that in the sort of expertise in your scientific advisory and working groups as well. Yes. So another question from Mohammed El Shabini, sorry if I've pronounced that wrong. So this sounds very ambitious. How does this fit with the Foreign Secretary or UK government plans in supporting LMICs with whole genome sequencing and creating a global biobanking database? There's also work being undertaken now with Africa CDC. So is there any coordination there as well?
Yes, as I noted in one of the slides, we will be working with other agencies that are in this space. We know Africa CDC is doing work. We know World Health Organization is supporting 12 countries in Africa on sequencing, and other agencies, BMBF, Swiss Biobank, FIND is already doing some work. And then if the Foreign Secretary for the UK has that great vision to do that, then we can become the vehicle to implement this in the LMICs.
Great. We've just got a few seconds left, so I'll just ask one quick question. Will there be a standardized way of sample collection, such as frequency of sample collection per individual over the course of infection?
Yes, so that's part of the protocol development. So in the proposal stage, we will make sure we agree with the partners on the standard protocol to follow, and what's the frequency, what type of specimens, what quality do we need, what minimum metadata should you collect on the sample. All that will be agreed at the proposal stage, because we want to make sure that there is standard procedures that are followed for the samples that are biobanked or taken for sequencing.
Thanks, Wilber. There's some great amount of questions, but I know we're running low on time. So I think if there are more questions, to be encouraged to use the form, and Wilber will be able to answer them at a later date. Thank you.
You're welcome.
Thank you very much, Wilber, and thank you, Divya. That was great. So now we're going to something different. This is supporting ethical reflection in COVID-19 research and policy. And we welcome our colleagues from the ethics working group of the coalition, in fact, 4 of them. Katharine Wright, who is the Assistant Director of the Nuffield Council on Bioethics. Professor Cesar Atuire, Senior Lecturer at the University of Ghana. Professor Oumou Bah-Sow, who is the President of the National Research Ethics Committee of the Republic of Guinea, and Elysee Nouvet, Associate Professor at the University of Western Ontario. I'm very grateful indeed for Katharine Littler from the World Health Organization, who's kindly agreed to moderate this session. So over to Katharine.
Thank you very much. And I'm honoured to be asked to co-present on behalf of the Ethics Working Group, and in particular on behalf of its co-chairs, Jennifer Ambe and Cheryl Cox-McPherson. And the next slide, please. So the Ethics Working Group brings together a wealth of ethical expertise, primarily from low and middle income countries. And our initial remit was very much about offering ethical advice on request when approached by researchers trying to think through how to tackle specific ethical challenges arising in their studies. And what we've learned, I think, over the last year is that the real need may be less for this kind of reactive kind of advice, although that may still have a value, but rather for finding ways more broadly to help reinforce and democratize ethical thinking and research during emergencies. And we suggest this plays out in at least 3 ways. Firstly, I think the very well-recognised need for better resource systems in many low and middle income countries to support ethical review and reflection. And this remains a challenge in many countries, despite some very valuable initiatives, such as that by EDC-TP. Secondly, in the importance of ethical thinking being embedded throughout the whole chain of research. It's not just the responsibility of ethics committees to sort out the ethics, as it were, at the point of review. It's actually a matter for everyone involved in research. And this is an important point for funders' own practices too. And then thirdly, in the need to find ways to foster better public conversations about the ethics of SANS. And we just touched on that briefly in the last Q&A about biobanking. If SANS remains by elites for elites, How can we expect people to trust it, buy into it, use its products, such as vaccines? Next slide, please. So looking at this big picture, as a group, we've identified as a starter 2 important research priorities for ethics. First, to think about how we make ethics the business of everyone involved in research, embedding ethics, as it were, in all our practices. And this will involve exploring, developing, and crucially finding ways to implement effective strategies to help ensure that the ethical conduct of research becomes an integral part of the whole research process in public health emergencies, from planning, design, to review, conduct, and finalization of studies. And then second, to look really critically at how effective existing community engagement practices for research have been during COVID-19 and to innovate new practices. Given the nature of pandemics and the associated social distancing and other public health measures, this should involve a particular focus on how technologies can support meaningful engagement. And pick up again on the comments at the end of the previous presentation, which I was delighted to hear. I think this kind of community and wider stakeholder engagement involving those who will be affected by the research as well as those doing the research is at the heart of ethical research. It's not an optional add-on separate from ethics. Next slide, please. So picking out one strand of these priorities that the experience and global reach of the members of our ethics working group will be well placed to deliver, we propose a global consultation on challenges, opportunities, values and practices in the effective ethical oversight of COVID-19 research, with the aim of mapping the gaps that need to be filled to support more countries to be more ethically prepared for future pandemics. This would involve engaging critically with the experience of ethics committees and other stakeholders to identify challenges and opportunities for strengthening research ethics capacity. And that should include promoting discussion of how ethical reflection can be supported throughout the whole research process, not just at the point of review. And then critically exploring which tools, resources, values and principles are effective for reinforcing capacity and confidence in those scrutinising research in emergencies, asking what works. And we'd envisage including, among other tools, consideration of the ethical compass, which was produced as a result of the Nuffield Council's work on ethics and research in emergencies, based around values of equal respect, fairness, and helping reduce suffering. So this idea has already been piloted, as it were, in some workshops in Guinea in February this year, as part of a partnership between Professor Bah-Sow of the National Ethics Committee in Guinea, Elysee Nouvet, a member of the Ethics Working Group, and Dr. Julian Sheether of the Nuffield Council Working Group. And I'd like now to hand over to Oumou and Elysee to talk about the Guinea experience. Thank you.
Thank you very much. I think I will jump back right in, given the restrictions of time. Oumou, are you there? Okay, so I will jump in. My name is Elysee Nouvet. Thank you very much, Katharine, for that introduction. And so Professor Oumou Bah-Sow has prepared a presentation in French, and given the time restrictions, I will be presenting on her behalf. So research on infectious disease outbreaks obviously raises serious ethical questions in resource-limited countries in particular. These can lead to serious harms, including violations of fundamental human rights, and leave a lasting legacy of mistrust and suspicion. So organizing workshops dedicated to collective reflection on our ethical challenges is of paramount importance in resource-limited countries. If these workshops are organized in several countries, they would allow for a fruitful exchange of experiences and strategies. We must take the time, especially during a pandemic such as the current COVID-19 one, to name, clarify, and compare our major ethical concerns in low and middle income countries so that they can be addressed and taken into account. I had the honor of co-leading such workshops in February 2021 in Guinea, funded by the World Health Organization and in partnership with the Newfield Bioethics Council, the National Health Research Ethics Committee of Guinea, and the Coalition Ethics Working Group. Guinea is a unique setting for COVID-19 clinical research, given its recent experience with an influx of research during the 2014-2016 Ebola outbreak. Bringing together members of Guinea's Scientific Council for COVID on day one and members of the National Health Research Ethics Committee on day two, We explored the utility of the new field ethical compass for deliberating on ethical challenges during this public health emergency, and had the opportunity to reach agreement on some of the more troublesome questions our country faces for clinical research, specific to the COVID-19 context. For example, we noted that during the 2014 to 2016 Ebola outbreak, the major suffering and fear caused by the epidemic was exacerbated by widespread mistrust of national and international actors, but also by a lack of community engagement, which was the source of unimaginable violence, fear, and psychological suffering. We agreed that core to the practical and ethical possibility of research in the country is community engagement. And while it has been central to the COVID-19 response since the outset of that response in Guinea, there remains much more work to be done to ensure that this is more than a checkbox approach to thinking about community engagement in the country, and to understand that really what is at stake with community engagement is respect for populations and the possibility of added psychological suffering when it is not done. We talked about the importance of scientific committees setting minimum standards for research during public health emergencies, something that's been done in Guinea, but could be clearer. Global inequalities in leadership and distribution of research benefits remains flagrant. Certainly, we know that we need to have more opportunities for workshops like those held in Guinea to be organized in as many countries of the South as possible to help identify and manage ethical challenges observed in health and public health emergencies, and to ensure we learn, are better prepared, and can put into action national and global commitments to ethical clinical research and community engagement. Thank you.
Katharine, could you continue for Caesar?
I will, yes, though I'm very sorry that Caesar can't present this in his own voice. So in brief, the ethics working group is very keen to build in the way that Elysee and Oumou just described on the Guinea experience in members' own countries and regions, with an aim both to promote and support local reflection on how to tackle the kind of challenges thrown up by the pandemic. And that's both in terms of challenges for ethical review and reflection in research, and also more broadly in related policy, looking, for example, at how vaccine access affects trust in vaccine research. And then to draw together common themes in the way that Elysee and Oumou described across countries and regions for future learning and implementation. Next slide, please. So possible guiding questions that could be explored in similar kind of workshops as the ones that took place in Guinea, but being alert to the fact that these might look different in different settings, would include what ethical issues remain unresolved in participating sites. By sites, we would think of either institutions, countries, or regions, depending on how these events were organised, that continue to present barriers to the implementation of a successful response to the pandemic. Then secondly, thinking about how such issues could be addressed, what guiding principles or tools could or should be invoked in order to support an ethically informed response to a pandemic. And that would include thinking, as in Guinea, about how the ethical compass of the three values might fit in this context. And then thirdly, I think some real learning across the sites, the benefit of having this kind of international global approach, looking at what ethical issues have been successfully addressed in the participating sites and what lessons other sites can learn from this. So next slide, please. So thinking about the scale of this project and possible timelines, interests have been expressed by ethics working group members and potentially by others in their networks in holding these kind of consultations, including in Latin America and the Caribbean, in Asia and Asia Pacific, and across a number of regions in Africa. And we're very interested to hear from others who might also be interested in being involved. We feel this is a really valuable opportunity to build on the experience from Guinea in a timely way while the pandemic remains a global priority. And also, I think, to build in ways that those particular discussions were with the Sounds Committee and with the Ethics Committee, looking as appropriate in different countries as to how other kinds of stakeholders might be involved. And when Caesar and I were debating this earlier, he was talking about the possibility of involving young people through social media, journalists and so on in the Ghanaian context. We then envisaged sharing common themes and findings through an international symposium or webinar like this, as well as through publications. And we know that the wider coalition and also the WHO Epidemic Ethics Network would also be well placed and be enthusiastic to help in disseminating those findings. Next slide. Thank you very much.
Thank you to all our speakers, and including Caesar. I'm sorry we couldn't get a better connection, and to Professor Bah-Sow for putting her slides together. We will head straight into the questions. I think this is super exciting to have both an ethics proposal and one that's building on a project coming out of a low and middle income country or a low resource setting as one of the 4 priority projects. So it just shows how important I think ethics is in emergencies and pandemics. So it's a great highlight. We're going to follow the same format. So please put your hand up if you've got a question. I'll try to come to you or type it into the chat. I see we've got a lot of comments, but it'd be great to have a question. Maybe whilst we're waiting for one, I'm going to kick in, and apologies if I can't see your hands. In terms of, you've made a strong, I think, and compelling case that there's really a need to embed ethics throughout the research cycle. In terms of, and your questions are broad, I think your proposed questions are to be able to do that. But are you going to focus on particular elements or particular audiences? As you said, funders have responsibility, research ethics committees, researchers. So who are the stakeholders you're going to engage, I think is my first question. And then I think I'll follow up with someone who's got their hand up. Who wants to take it? Katharine, do you want to kick off? Or maybe Caesar? I don't know if we've got Caesar. It'd be great to hear from you if we can. Caesar, let's try get you connected.
I'll speak to me. Clear.
Still cutting out. Maybe type.
All right.
Katharine, did you want to kick off, and then maybe Elysee?
Sure, yes, and then hopefully, as you say, Caesar can pop some things into the chat. So I mean, it's an excellent question, Katharine. Thank you very much. I think in terms of what would come out of any individual country or regional workshop would very much be determined bottom up, in the sense of having a real sense of who the key stakeholders are in that case. And as I say, when we talked about this earlier, and Caesar was talking about what the Ghanaian experience might be, he felt younger people and journalists were a particular audience there, and I think that picks up with some of the wider discussions about communities. And following the model of the Guinea workshop, that was very much about starting with what were the challenges and then thinking about how they might be met. So I think that is that sense of being bottom up. And I think there is possibly another parallel piece of work. I mean, other things that I've been sort of following with a Nuffield Council hat on, in how one could explore with funders some of the questions around, how you embed the idea that you're ethically responsible into how you submit calls, what you ask for in applications, how you review applications, who's involved in doing them, what the values are that undermine that. So I think that there are so many different possible strands. This particular presentation was trying to put forward one particular one, so country by country, and I think my quick answer to that will be determined on a country by country basis.
Thanks, Katharine. We've got a hand up, and I'm going to apologise in advance for the pronunciation of your name, but Notom Bizardwa Naoni. You have your hand up. Would you like to ask your question? Unmute yourself and ask your question. If not, I'll move on to one of the ones in the chat. So Irene, we've got a question from Irene, which is, do you plan to explore aspects of social value of COVID-19 research at a time when community suspicions on the research enterprise have been heightened?
I think the answer has to be yes, but could I hand over to Elysee perhaps to reflect on how those questions emerged in Guinea?
Yes, thank you. So one of the things that is really made Guinea an ideal site for this initial consultation is that there has been a great deal of reflection and scholarship and documentation and surfacing of the ways in which so-called resistance to clinical research during Ebola and during the West Africa outbreak was actually, very clearly articulated to histories of distrust and practices and distrust of national and global actors. So for every context, there has to be a community engagement strategy that really is informed by that deep knowledge of the context. So for sure, we expect that these consultations will be a means to clarify the ways in which ethical principles get put under additional pressure within specific settings based on those kind of social particularities. And scientific and ethical committees do have community representation as well as social scientists on them in most cases. So they bring to the table, and certainly did in Guinea, those perspectives and that in-depth knowledge. Over.
Brilliant, thanks Elysee. We've got a question about, will you be looking or interacting with field workers? I don't know if you did that in the Guinea project, but going forward, are they part of your core constituency of people you'll be looking at as well?
Again, I think exactly how this works out will play out differently in each country, but I would see them as being an absolutely key part of the process. Could I just say, Katharine, Caesar's popped something in the chat. I think it's just gone to panelists rather than everyone. Perhaps you could read it out in terms of the response to the earlier.
Yes. Thank you very much for drawing it to my attention. So Caesar has put, we will broaden to think and engage with not only institutional and traditional community stakeholders. There are many other groups that are important: media, young people, social media. And these influence trust in science and research. So I think in essence, that's responding to multiple of the questions, as we're saying, about the changing interface of how people see research in the COVID setting. We've got a very practical question. I think one, how you're going to work with other working groups. Obviously, there's interest from the social science. I presume this would be a cross-cutting. You're nodding, so I can see that. And then there's a very practical question about harmonization of the protocols and how you're going to resolve those sorts of linguistic attention issues. But I'm assuming some of you have done cross-country research before. And actually, Elysee, you would have had that with translations of materials in the Guinea one as well.
Yeah, indeed, it's crucial to have somebody on the team who has people on the team who have the ability to verify the cross translations and budgetary lines for translation. The report from the Guinea consultation was made available in French and in English. Certainly we would that's a huge, linguistic barriers are actually a huge barrier for ethical global health research. So it's something we definitely want to ensure is addressed and taken into account in the budgets and planning for these consultations.
Fantastic, thank you. So there are a couple more. I think I've got one that's kind of a summary question. Once again, to your point about ethics being a key facet, but what do you think would be some of or from your experience today, will be some of the additional layers of complexity to do with health emergencies in terms of the ethics and some of the additional challenges? And how do you intend to bring that in to this work? And while you're thinking about that, Caesar's put another response in, so I'll read that out so everyone can see it. An important aspect is also seeing how to help ethics review committees gain more independence from institutional and political pressure. And I think that is in response to one of the questions in the chat that talks about the lack of separation of powers that has occurred in the past with ethics review committees not having any independence from maybe some of the state mechanisms. So I think that's something that you can target. But back to that question about some of the challenges that you might see that are unique to the emergency context and how you want to work on that. And then I think we've got about 1 more minute left.
If I could just respond briefly to that, Katharine, I think, and it came out again in Oumou and Elysee's talk, I think one of the things that is not necessarily unique, but more extreme in an emergency, is that sense of threat, of time pressure, and the sense of, on the one hand, it can't be the case that everything and anything can go, that we can just do what we like because it's so urgent, but there can be the pressure to feel like that, and there can be political pressure to get moving with things. And on the other hand, you don't want to be having, we have to do 6 or 8 weeks to do this because that's what we've always done. So how you adapt procedures appropriately. But I think also beyond that, I think it's recognising actually the human toll that emergencies take on us as individuals, as researchers, as those involved in scrutinising research and potential participants. The actual emotional part. I think historically ethics has been very bad about thinking of people as people with emotion. It focuses very much on rational human beings following principles. I think some understanding of actually the impact emergencies have on all of us in the emergency process. It's certainly something that came up very clearly in the Nuffield Health input that we had, the input we had to the Nuffield Health project. And I think that's something I would expect, but obviously can't be certain, would emerge in some of these conversations as well.
Fantastic. Thank you all. I think our time is up. There is one final question in there about, is enrolling patients in study design without adequate power and poor design an ethical issue? And the answer is yes. And maybe that is something that this group can look into when they're doing their work. And I apologize if I didn't get to all your questions. And thank you to all the panelists.
Thanks, everyone, for participating in that really useful and interesting conversation. So our last presentation is on excess mortality, alternative methods for evaluating excess mortality in low resource settings. Very pleased to introduce Professor Philippe Guérin. He's a fellow member of our steering committee of the coalition, and he directs the Infectious Diseases Data Observatory. And our second presenter is Dr. Professor Swarup Sarkar, formerly the C.G. Pandit National Chair at the Indian Council of Medical Research and currently working in the University of Göteborg in Sweden. And they will be joined by Dr. Marc-Alain Widowson, who directs the Institute of Tropical Medicine in Antwerp, and he's going to moderate the session. So over to you, Philippe.
Thank you, Nick, and good afternoon, good evening, everyone. It's a pleasure to present on behalf of a larger group this problem of excess mortality in low resource setting and how we could tackle that. And I'd like to acknowledge Veronika and Akona Changela from the Africa CDC who has helped us putting together those slides. I'm trying to, next slide, I can't control it, it seems. Thank you. So why is excess mortality important? Well, the impact of the pandemic is more than the number of cases and deaths reported by countries. And although we are seeing a lot of that, there is a lot of indirect impact, which are connected to the fear to access to emergency services, delay to access to healthcare services, including antenatal care visit, disruptions, among other things, of routine immunization campaign, malaria, HIV, and TB control program, and disruption of food and medicine supplies, exemplified by the lockdown measure. And so evaluating all causes of mortality patterns and trends can gauge the full extent of the pandemic impact, and this is important for this pandemic, but also to learn potentially for the future, but also help immediately address indirect impact of the pandemic. Next slide. The World Health Organization has made an assessment of the situation about data which could help us gathering and understanding the excess mortality. And their conclusion was that there is a significant data gaps existing in the African, Eastern Mediterranean, Southeast Asian, and Western Pacific regions. And it's illustrated by this map where in orange, this is the places and the country where excess mortality data are not publicly available. And only 16 of the 106 member states in this region have sufficient data to make empirical calculations. So there is clearly a problem which is mostly affecting LMIC to gauge excess mortality. And next slide. I leave the floor to my colleague, Dr. Sarkar.
Thank you, Philippe. I don't know whether I'm visible, but I'll try to elaborate on the case of excess mortality, taking an example from India. And my point from this slide is, it's not only a neglected area, it's a priority research area. You look at the just released publication list anthology from Indian Council of Medical Research, the apex body of research in India. Yesterday, it documented that there has been more than 200 publications by Indian scientists from one institute alone. And this is all over the world, Lancet, BMJ, Nature, New England Journal, everywhere. But if you look at the mortality study, none exist in these more than 200 publications. And there are 4 stalwart statistical and demographic institutes in India. I have listed few. Register General of India, which carries out, billions of data every year for billion population, they exist very well, and they run longitudinal and cross-sectional survey on model and national sample survey. National Statistical Commission is there from another ministry. Indian Statistical Institute has globally contributed on statistical methodology. The DHS, NFHS is conducted by Indian Institute of Population Science. In spite of these 4 stalwart institutes, and with an experience of very competent scientists at a global level, we still depend on the New York Times that has only published this data, where that death range, you see that, that it varies from 300,000 to minimum of two times to, a maximum range of 14 times. So the point is, it's not only a neglected area by a very competent country and a competent scientist group, but at the same time, it's a priority area, because unless these numbers are owned by the government and the people of a country, it is not going to change our future preparedness. Next slide, please, Philippe. You'd see that, this, although our aim is mainly to measure the global impact of or help the countries measure the global impact of the pandemic in the low middle income country, we are not trying to spread everywhere. We'll be focusing mainly on the countries where there is a less resilient health system. For example, in many parts of India, parts of Africa, high populous countries, in Southeast Asia, deaths and births are even registered, to the extent of 10 to 30% of deaths and births are under-registered. So these kind of countries, we can easily expect there will be huge underestimation of mortality. Now, the purpose of this is not to carry out the research for the country, but to supplement and not to replace WHO or IHME mechanism which exists, or the country's own national mechanism, but to supplement them. At the same time, we cannot just depend on the big survey to be conducted and finished after 3 years or 2 years of time by the national governments. We also need local planning and local use of data. And that's how my third bullet on the second point is, we need an innovative approach where immediate local data can be generated and people can use it for the next wave to come, to prepare for beds, prepare for ICU, and even to mitigate socioeconomic impact for the families who had faced catastrophic health expenditure. And the last but very important point is, North versus South. There's a lot of questions always about access to the data by everybody, and the recognition and the benefit to the local people. And that's how the purpose of this work would be to support local researcher and see that they get recognized, they can publish in their local journals, as well as it can be comparable to other studies, so that there is a congruent one set of data and comparability. Next slide, please. And the purpose, it should be done through local and regional hubs, because purpose is not just to carry out a research, but to see that the research has an ownership and that data is used locally. And that's where, we have to look for people who are willing, and the researcher who are looking for a design, who are looking for whatever support they might require. And then can we network them with other global and North-South researchers, And then at the same time, can we work with the governments to create advocacy and to see that, the government recognizes need for this and allows the different researchers to carry out data. So we want to both enable researchers by advocacy with the government, as well as we want to empower local researcher by technical support and, when necessary, financial support. And, my most important point is this data is for local use. And that's where, waiting for regional and, big surveys is not enough. We need to quickly come out with even local methods, innovative methods of, which we have used largely on HIV, capture-recapture for measuring stigmatized diseases, or say, stigmatized numbers on mortality, addiction, snowballing, multiplier, many methods we have used where data did not exist, To begin with, we had only 4-country model in WHO when we projected for 170 countries. So such things are possible where local data can be generated by innovative methodology and people can use that. And the last important point is many people starting from last year, January, when the disease was not known, test was not known, have died without knowing they have died from COVID. In the mature epidemic in India, many people died just because of a lack of a bed or an oxygen, The minimum thing we can do today is to show respect to them, to let people know that their death have not been in vain. At least they have been counted, they have been measured, so that the future pandemics, they have contributed in measuring our possible impact for a future pandemic preparedness. Over to you, Philippe.
Thank you, Swaroop. Next slide. So the expected outputs of this work would be first, a short term, present the first estimate of all-cause mortality patterns and trends derived from LMIC region data sources, valorize data which are existing, and we know that there are several groups which have done that at community, regional, or municipality levels, identify the needs and data availability and accessibility gaps in LMIC. We believe also that this project could provide, on the midterm, link to other sources of information, estimate mortality patterns in vulnerable population subgroups, and I'd like to echo Swaroop's voice on looking at healthcare workers, migrant population who are often even more neglected, and develop data quality tools for quality improvement. The long-term plan is very much what WHO and the African CDC are planning into establishing regional data hub. And so we are not duplicating that, but we would be addressing earlier priorities. Next slide.
1 minute.
The slide has been shared by our colleagues from the Africa CDC. So just highlighting that the Africa CDC is putting in place resources for putting together rapid mortality surveillance program moving forward. But we know that this effort, although extremely important for the future, will probably not be able to release data in a very short period of time. Very critical, but what we are proposing will be complementary to that effort as well as the effort of WHO. Next slide. And so the proposal is very much, we don't think that there is a single organization in the world right now that can lead that on its own. And it's probably rather using the coalition and a network of regional initiatives, making sure that we standardize this approach. There is a huge issue of standardization on excess mortality, as many other things that we have heard earlier on. And this will be a joint effort between the data sharing, the clinical epidemiology, the data management, the ethics, and the social science working group to come together and help us pulling this critical data that will not only help the country, but help the response to this pandemic. And thank you very much.
Great. Thank you very much, Professor Guérin and Dr. Sarkar. That was great, and really such a tremendously important topic. I mean, gathering appropriate mortality data in a timely manner really is what drives intervention development and implementation, sustains control programs. And it's so incredibly difficult to do, particularly in settings which perhaps need it most. And that's been a consistent challenge. And I think perhaps even more difficult to do it in COVID, where in many ways, some of the interventions are actually reducing mortality for other causes, and then that even confuses it even further. So we have a couple of questions, one question in the chat. Before we turn to that, I'll just start off with a question of my own, which is, because of the challenges that we come across for gathering routine data on mortality from countries, how do you see this effort kind of feeding into a kind of sustainable long-term effort so that we can get timely mortality data to sort of notice signals of mortality and to understand diseases as they happen? How do you think your initiative will feed into low and middle income healthcare systems and vital statistic gathering systems with governments that will strengthen that in the long term?
So, I leave it to you to answer that question.
I think, the very much, capacity building that even during emergency, you can collect nearly close to the, valid data is a very important exercise for the countries to know, and the use of it. But unless you know the dates now, you cannot prepare for the next wave or the preparedness of the health system. But on the long term, I think, long term, it's a complex issue about where even birth and deaths are not registered, even with the highly competent countries, or even in a political system, where often, even there is a, what you call, what would be the right term? Denial of death, due to a particular disease. That happens in all the epidemics. We have seen this. People who work in epidemic, they know. So I think we have to have this immediate capacity building, which will create the confidence that even in emergency, you can collect such number. That's a very big step, I think.
Great. Thank you so much. And it kind of related a little bit. We have a question from Baba Sahib Tandali. Again, apologies for pronunciation. And the question is, for mortality estimations, how could the Sentinel community approaches be explored, considered, and utilized in LMICs? And I guess this relates to, some groups have been using, verbal autopsy and DH in demographic surveillance systems, HDSSs, to sort of understand this, which I guess is and extrapolating to regions. Can you speak a little bit to that, to the Sentinel community approaches?
Let me answer this, because I have personally used this when first time we saw HIV epidemic in 89, late 90s or early 90s. In small areas where no health system exists, we could even just count the new on the burial ground, on the new burials. And for a local city, we could measure how many deaths have happened. And we could extrapolate this by using multiplier, and we could just periodically do it every month, a year, and for several years. So there are very innovative methodology if you engage the community and make community understand that it is for their interest. You will see enormous response. And today in the Facebook method, I could see that there is a huge potential for using local data for national and local and district level. Over.
And if I may add to that question, I don't think that there is one method that we could apply for the world. There will be some countries, and we have had partners in Liberia, for instance, from the Public Health Institute, saying that they are really interested to participate in that. And in that case, that's the national central government which wants to participate in that effort. In other countries, that's going to be census. In other countries, that's going to be burial counts. So I do think that there is a patchwork of data that we can use to get to this estimate. And I think maybe the mistake would be to try to imagine that there is a unique way of measuring that. It's not the case. And that's going to be this innovation of multiple methodology coming together.
Yes, that's great. I think from personal perspective, I think that's very true with influenza. We had the same challenge. And I think that triangulating, they're all wrong. All the estimates are wrong, but by getting more different techniques and different estimates, we can sort of start to hone in on what may be going on. And perhaps there's another question here, but I think it's more of a generic question, and I don't know whether it's more about the whole session rather than this particular one. So perhaps we'll just continue on the line of this on excess mortality. Which is, you mentioned complementarity to HME and the work that they're doing. Could you kind of highlight a little bit more that difference between what IHME is doing and what you'd be proposing?
Well, I think IHME has worked very closely with the governments to extrapolate their data, which are existing, the official data. In other places, they are part of what we are providing. But I think, considering the scale of lack of data in excess mortality, we need to go one step further and use more efficiently the research community. Again, the official excess mortality data have been usually coming from public institutions. Here, I think our approach is it's a bottom-up approach, coming from the research community and generating the data and making the data available for making that exercise possible. And so it's complementary again from IHME approach as well as WHO and the African CDC. And I think that's very much in line with the complementarity and triangulation that you were referring to, Marc-Alain.
Fantastic. I don't see any more questions in the question and answer nor hands up. So we've only got less than a minute. So I think we'll end it there with my thanks to both presenters for really such an important topic, and hopefully we can move ahead with this. Thank you very much, and over to Professor Wright.
Thank you very much, Philippe, Swarup, and Marc-Alain for that really interesting presentation. So now it gives me great pleasure, and it's a privilege to welcome Dr. Soumya Swaminathan. She's the Chief Scientist of the World Health Organization. She's going to speak to us about the WHO's perspective on prioritizing research in low resource settings and public health emergencies. Over to you, Soumya.
Thank you. Thank you very much, Nick. And greetings to colleagues who have joined this meeting. I see a lot of familiar faces, and apologize for not being with you throughout the session. It was, when I saw the agenda, it was really very fascinating, and I wish I could have joined. But I'm really happy that this group has come together, and the Clinical Research Consortium that was formed, last year, and is really moving ahead, is vibrant, and is discussing the issues of concern. One of the things, of course, that the WHO is very keen to promote is the facilitation, coordination, promotion of the research priorities, and we did that from the very beginning of the pandemic through the R&D blueprint to prevent epidemics. And because it was something that had been set up, after the Ebola outbreak, and that it had been functioning, of course, it has a focus on product development for priority pathogens, and developing a research roadmap, and developing target product profiles, and then laying out the protocols that would be needed to do that. But through the experience that was gained in Ebola, but then also in Lassa fever in West Africa, and Nipah in India and Southeast Asia, it was very clear that you need a model which really empowers the researchers who are in those countries that are facing these problems, on a recurrent basis. And that top-down driven research is not going to be as effective as really problem solving. And what I could see was that the approach taken for Lassa with leadership in Nigeria, particularly by Dr. Chikwe and his colleagues, really bringing together the countries of West Africa which suffer from Lassa, starting with the very basics, like developing the laboratory capacity for diagnosis, which you could see then in subsequent years, the cases diagnosed increasing, for developing local epidemiology and documentation of infection, which is such a basic thing, and then also developing studies which looked at transmission in the community, considering both the zoonotic host, the rodent host, as well as the humans, as a basis for being able to test diagnostics and vaccines when they eventually become available. Because without this type of a basic network, infrastructure, and data on the disease and its profile in that region, it's very difficult to bring in a new diagnostic or a vaccine and set up high quality clinical trials to test it. So really underscoring the importance of sustained and locally led research initiatives in order to really have an impact in the long run. In fact, we've all seen now I presume everybody's seen the report that was submitted by the Pandemic Public-Private Partnership, the PPP, the 100-day mission document that went to the G7 leaders, really talking about the need to prepare for the next pandemic, and basically focusing on R&D. How do you get therapeutics, vaccines, and diagnostics developed within 100 days of the declaration of a pandemic? So again, the focus there is on product development, on how the public and private sectors can work together, and and importantly on ensuring equitable access to those products. And we know that unless you think about access from the very beginning of research, it's not going to happen. If it's an afterthought or a consideration that comes later, and despite having had the ACT Accelerator and COVAX set up exactly to ensure equitable access to vaccines and other products, we are still facing a situation of a high degree of inequity. So there's a lot of work, I think, to be done on the existing R&D ecosystem and the way that investments lead to equitable access, in the sense that the balance between public investments in research not actually leading to the same public benefits or public good is something that definitely needs to change. So we really also welcome the efforts to this type of effort on the research coalition to focus on the research needs of low and middle income countries, because the context is often very different, as we know, from the developed world, and many of the guidance documents really need to be considerably adapted before they can be adopted and implemented. And I often ask myself that question when I see a guidance document. Of course, WHO speaks to the whole world, but our major audience is the LMICs and the LICs. And so any guidance that we put out, we have to think about how is this going to be implemented, and is it relevant, and will it change from context? And of course, yes, the answer to all of that is yes, and it needs local implementation research in order to determine the best approaches. So we also know from data that was presented by the GLOPID-R network of funders on infectious disease research, for example, when we had our last global forum on COVID research, that out of the 9,000 or so research projects that have been funded in the last 1.5 years by these 20, I believe it's 27 research funders, that less than 1,000 were actually done in the LMICs, and a very small proportion of the overall research budget was actually being spent in LMICs. So there is a very big imbalance here, and which is again something that we need to continue to correct in many ways. I think one is really advocating for local governments to invest in research as well, because I think unless the local government ministries of health and other ministries really feel that research is providing inputs into their policy making, is beneficial, is providing rewards, they are not going to be really incentivized to invest in it. And I think a good example now is when we are faced with these vaccine supply shortages, that many countries now want to invest in vaccine manufacturing. They want to invest particularly in mRNA technology, because they see that countries that have invested in R&D, in manufacturing, are now actually having an advantage because they have access to vaccines. So I think it's things like that, but it's on the policy side, it's also important to involve the health policymakers and the end users of research right from the beginning. And this is a lesson I learned as well in my career as a researcher, that policymakers accept much more when they've been involved and consulted from the beginning. And in fact, when they help to shape the research questions, rather than when you go to them with a finished product, almost telling them, to use your findings or to change policy based on what you found. So I think the approach that researchers take also has to change, particularly when it relates to something that could impact on policy. I understand that there were 4 areas that were discussed today by the working groups, and there is indeed a need to focus on these areas. The characterization of long COVID in LMICs, again, we saw that most of the published data, we had a big consultation yesterday on long COVID, and most of the research, most of the documentation on long COVID has come from high income countries. And we really need to look at this in low. So this is really an important topic, both in adults and in children, across a diversity of ethnicities, race, age, sex and socioeconomic living conditions. I think it's really important. The biorepository capacity in LMIC is also very important, something I've struggled with personally as a TB researcher. When there is a question that comes up, or there is a company that claims to have a good diagnostic test, unless you have a biorepository to test that with, you waste a lot of time, and then you are dependent on having it tested outside in other countries. So really important. Of course, again, it's an investment. It costs money to run a good biorepository, but perhaps this is something that could be thought of regionally or sub-regionally rather than setting up one in every country. The reflections on ethics, particularly around COVID research and policy, again, is really important. And I've been having discussions with our own ethics review committee here to think about how, in a pandemic, these large multi-country studies which adopt a common protocol, what kinds of ethics reviews and approvals we should put in place, as well as regulatory reviews and approvals, so that we're well harmonized. We don't spend a lot of time in getting these approvals, and we're able to move ahead. So all of this needs to be done, obviously, during peacetime and the inter-pandemic period, in order to have these kind of policies in place which can work during an emergency. And then as I came in, you were discussing the new approaches to measuring excess mortality. Again, a very important topic. We see data now coming from many countries which show that the documentation of deaths is clearly an underestimate of the true excess mortality, both due to COVID and due to other causes of death, which I think for LMICs is going to be very critical to understand, because we need to know if the deaths due to TB and malaria are increasing, if the deaths in antenatal and postnatal women or under 5 children are increasing. All of these are going to be very important for the policymakers to reflect on and to quickly take corrective action. So while it's a sensitive topic for many governments, its importance cannot be underestimated. And I think discussion on methodologies which can be done in LMICs again would be, add a lot of value over and above the modelings that have been performed. And finally, I mean, I just have to say that the importance of global solidarity cannot be overstated. We have seen during the pandemic that the only reason that science was able to advance rapidly and provide all of these new solutions and new tools is because of the global collaboration that we saw, the willingness and openness to share data, knowledge, information, right from the sharing of the first genome sequences from China to the open sharing of all new knowledge generated in the archives, and the fact that journals also made all COVID-related research open access. So I think these were all very good signs, and they give a pointer towards things that could be done differently, even in a non-pandemic situation. When you're faced with a big global public health problem, it could be NCDs, it could be TB, it could be anything. If there is this mindset to solve the problem, then clearly this global community of researchers and scientists are up to that challenge and can do it. So it's important for us to work together and also to communicate and coordinate our efforts. This is where WHO's role, I think, comes in, so that the efforts of different groups are complementary and not duplicative. We've had several discussions with the other trial platforms, for example, outpatient and inpatient trials, to see how we could have done better, or we could still do better in terms of prioritizing the drugs that need to go into trials, or working with existing networks of clinical trial platforms. And indeed, there is a need to strengthen and develop more of these kind of networks, I believe, at the regional level, which in the inter-pandemic period will continue to do trials, because that's the only way to develop a cadre of well-trained and well-qualified people, is to keep on doing trials and be very conversant with GCP and GLP and all of that, ethics and regulatory issues. So this again needs sustained financing, but it will pay off a lot in the long run, because the rewards, we all know that, I think here on this group, I'm preaching to the converted, but we need to put that message out loud and clear that investment in research capacity, investment in clinical trial platforms, investment in R&D, and in the links between academic sector and the private sector. Of course, not forgetting NGOs and civil society who play an important role in community engagement and advocacy. All of these are really important elements, and I would welcome any feedback from this group, in fact, to the WHO, to see if we can be more helpful in some way or do things that would help advance the work of this coalition. So thank you very much for giving me this opportunity to come in and say a few words, and I look forward to the outcomes of all of these areas of work that you were discussing. Thank you.
Thank you very much, Soumya, for your clear and important messages, and also for your realistic and insightful perspectives. We're very grateful. Thank you. It's now my great pleasure to introduce Professor Charles Wisonge. Charles is the director of the Cochrane Collaboration South Africa, based at the South African Medical Research Council, and he's also vice chair of GLOPIDA. Charles, over to you.
Thank you so much, Nick, and thank you to the coalition for inviting me to give the closing remarks for this wonderful meeting. So I am a vice chair of the Global Research Collaboration for Infectious Disease Preparedness, known as GLOPIDA for short. GLOPIDA has 29 members and 3 observers, and it is the only alliance of its kind to bring together research funding organizations on a global scale to facilitate an effective and rapid research of a significant outbreak of an infectious disease with epidemic and pandemic potential. LMICs are a top priority for GLOPIDA for COVID-19 research, as well as research capacity on any pathogen with epidemic or pandemic potential. At GLOPIDA, I chair a working group focused on research in low and middle income countries. Initially triggered by the COVID-19 pandemic, this working group has been created to support a coordinated approach to research funding towards epidemic and pandemic situations in low and middle income countries. Today's meeting is therefore very relevant to our work, and I thank everyone for the interesting presentations and rich discussions. The 4 presentations we had today support the WHO research roadmap for COVID-19. In addition, they have been identified by researchers in low resource settings as key priorities that have not yet been adequately addressed. In these and many ways, what we heard today illustrates all 7 principles for funders developed by GLOPIDA and the UK CDR as part of the COVID Circle collaboration. The first principle is to support funding that is aligned to global research agendas and locally identified priorities. We must support locally and regionally identified research priorities, even as we ensure that they contribute to the priorities set out by WHO, which are also based on input from researchers from around the world. The second principle is to support rapid research by building on existing capacity and supporting capacity strengthening, as seen in all 4 proposals that we have presented today. The third principle that we call on funders to support is that of equitable and inclusive research partnerships, particularly when research collaborations are between partners in the global south and in the global north. Open science and data sharing are clearly challenges faced by all of us during this pandemic, which may pose obstacles to the execution of some of these projects, but may also be tackled by the biobanking and excess mortality projects. Protection from harm and appropriate ethical consideration are the responsibilities of all researchers, and it has been a heavy and difficult responsibility for ethics committees reviewing COVID-19 related protocols under time pressures and sometimes without the expertise or experience to review the types of studies that have been put before them over the past year. I am therefore heartened to see a proposal for an exploration of the challenges faced by ethics committees in low resource settings, which I must say is badly needed. We must find a way to balance appropriate ethical reflection and protection of populations while responding rapidly to public health emergencies like the ones we are faced with now. Lastly, the principle of collaboration and learning enhanced through coordination is embodied in all four projects that were presented and discussed today. We have had good examples of South-South collaboration. In addition to North-South cooperation, the coalition has enabled a remarkably varied and multi-country set of working groups who have been active in discussing together the research gaps in their respective regions. I hope that the questions and discussions today will be helpful to the project groups as they refine their approaches and prepare to develop full proposals. I also hope that there will be ample opportunities to support these projects in the very near future, as these needs are great and these questions are urgent. Permit me to thank you immensely again for the wonderful meeting. Thank you.
Thank you very much indeed, Charles, for those wise words and those closing remarks. So it just leaves me to thank everyone, the keynote speakers, the presenters, the moderators, the funders, and all the interested researchers who participated in this meeting. We know that you're all busy people, and we appreciate your time and your interest. It's also rather an unusual meeting in that it didn't overrun. So I hope it's been useful, and I hope it's been informative, and it'll provide support and some momentum for the initiatives to address these important areas effectively. So finally, with that, on behalf of the COVID-19 Clinical Research Coalition, thank you very much, and I wish you a good day or a good night. And a good night. Goodbye.